Okay, ma'am. Am I audible, ma'am?
Yes, you are audible to me.
Okay, ma'am. So, good morning to all the participants. Good morning, Dr. Sumandata, ma'am. And also, good morning to our course director, Dr. Raman Silva Kumar, sir. So, this is Nadeem Riaz program, assistant of 21 days National Refresher Course, come capacity building program. And today's morning lecture is CRISPR-Cas genome editing principles and applications in horticultural crops. And the lecture will be delivered by Dr. Suman Lata, who is a senior scientist, agricultural biotechnology, Division of Vegetable Science, ICAR, IRI, New Delhi. So I will tell a little bit about Dr. Suman Lata, ma'am. Dr. Suman Lata has joined as a scientist at ICR, Indian Agriculture Research Institute, New Delhi, in the Division of Vegetable Science in 2016. She is a CSIR JRF Fellow and completed her doctoral degree, PhD, Molecular Biology and Biotechnology from the ICR, Indian Agriculture Research Institute, New Delhi. She is presently working in two crops with okra and tomato in areas of genetic improvement, genomics, and genome editing. She is a recipient of the Young Horticultural Science Award 2023 by Society for Horticultural Research and Development (SHRD). She has published 35 research papers in Journal of International and National Reputies. Thank you so much. Now I request Dr. Sumalata ma'am, please start your presentation.
Thank you, Dr. Nadeem, for the opportunity and a nice introduction. So, good morning to all of you. So, as mentioned earlier, the title of the today's talk is CRISPR-Cas genome editing principles and application in horticultural crops. So, in this lecture, I'm going to talk about the basics of the genome editing, the principles involved, various kind of the endonucleases which are used, and what are the applications of the genome editing. So, first, what is genome editing? So in genome editing, we talk about the technology which can produce genome modification, like targeted mutagenesis that we are doing here, mutations, but that is targeted at a specific site. So in this, we can do the insertions, nucleotide insertions, where the nucleotides like A, T, G, C, they can be inserted. Similarly, deletions can be created in the genome. Deletions when the nucleotides they get deleted. It can be one nucleotide and it can be many nucleotides and substitutions. So, in through genome editing, uh, the site-directed insertion, deletions, or substitutions can be made. They are targeted. It means they are at specific site. And we use the DNA for genome editing. The template is the DNA. And the genome editing relies on the production of the site-specific double-stranded DNA break. So this double-stranded DNA break is very important for genome editing. This break has to be made, and then this break has to be repaired. So the cellular machinery has its endogenous repair mechanism through which it repairs any break which occurs in the DNA. So in a normal day-to-day routine process of the cell, the DNA face many breaks and they get repaired. Similarly, if we introduce any break in the DNA through the site-directed nucleases, so these breaks are repaired. But the point here is These repairs, they are error prone. So when these repairs are made, so mutations are created. So there are two mechanisms of the repair. One is NHEJ, that is error prone non-homologous end joining. Non-homologous end joining, NHEJ. And the other method of repair, that is error free, which is homology direct repair, HDR. This method is error free. So we can see here, this is the DNA strand, and we are making a cut here. This is the site where we want to make a cut. So we can use any site-specific nuclease. So it can be ZFN, it can be TELN, it can be CRISPR-Cas9, or it can be CRISPR-CPF1. So any nuclease can be used. So the nuclease will make a cut here. So a double-stranded cut is made. Then this cut is repaired through the repair mechanisms. So the repair mechanisms, there are two repair mechanisms we discussed in the previous slide. One is NHEJ, that is non-homologous end joining, and other is homology direct repair. So when this break is repaired, so there are chances that some deletions will be created. Although the fragment is repaired, but it is not repaired uh, error free. Means during repairing process, some errors in the nucleotide happens, which created deletions. There are chances that during the repair process, some nucleotides may get inserted. So you will get insertion. So these are the DNA deletions or insertions created during the repair process. So gene knockout or disruption of DNA elements can take place during the repair process. The other mechanism of the repair is the homology direct repair. So in this repair process, we use the donor DNA. So this is the donor DNA, which is represented in this magenta color. So using this donor DNA, the repair process is taking place. So this is precise. So this is used in case of gene knock-in and gene replacement. Okay. So this is error-free process, but NHEJ is error-prone. But most commonly, this process is operated in the plants, NHEJ. So in plants, double-stranded breaks, they are mainly repaired by NHEJ. And this mechanism operates throughout the cell cycle in higher eukaryotes, and it shows low fidelity. Low fidelity means accuracy is low. And due to this error-prone nature, NHEJ leads to the addition or deletion of the nucleotides. And due to this, we can get complete loss of gene functions because indel introduced in the exons, they can lead to the missense or nonsense mutations. And as we've seen in the HDR, we use the homologous sequence. So this HDR, it operates during the S and G2 phase of the cell cycle, and it has much lower efficiency. So it is more challenging in case of plants. But this NHEJ process operates throughout the cell cycle in the cells. Okay. Now, what are the endonucleases which are used to produce site-specific double-stranded breaks? So, you have seen this that we are making a break. So, to introduce this break, there are many nucleases. So, what are the nucleases? So, one is meganucleases. Then there are zinc finger nucleases. Third, there are transcription activator-like effector nucleases, which are commonly known as TELNs. Then we have clustered regularly interspaced short palindromic repeats, which are known as CAS, and this CRISPR and the protein is the CRISPR associated 9, that is Cas9. So we have CRISPR Cas9, then we have CRISPR CPF1. So in this CRISPR, the enzyme is derived from the bacteria Previtola and Francisella. So this is known as CRISPR CPF1. So these are the various endonucleases which are used to introduce site-specific double-stranded breaks. So the meganucleases or homing nucleases, they are the first class of nucleases which were used for genome editing. And each nuclease or any enzyme, it has a recognition sequence, the DNA sequence which it can recognize. So for them, the recognition sequence was 12 to 40 base pairs. But because they were not used so much in plants, because the recognition sequence was very long, it has rare occurrence in the genome. So these, although they were first class of the nucleases, but they were not used so commonly in plants. After meganucleases came the ZFNs. They are the first generation of the genome editing tools, which were first exploited in plants in 2005. So, so these ZFNs, they are created by fusing the zinc finger. There are two zinc fingers, one zinc finger in the forward strand of the DNA or the target region, and one zinc finger for the reverse strand. And it has the enzyme FOC1, which acts only when it is present in the dimeric form. So this is the organization of the zinc fingers. So so the zinc fingers, individual ZFNs, they contain usually 3 to 6 zinc fingers, and they can recognize 9 to 18 base pair long DNA sequence. Okay, so zinc finger nucleases, they have a specificity through the zinc fingers. The nucleus is the FOC1, dimerized FOC1s, and when the cleavage is made, they create sticky ends. But the problem with the zinc finger was their design and assembly was technically challenging, and outsourcing was expensive. So these were the shortcomings which led to the limited widespread adoption of this nucleases for the scientific community. So they were used, but they were not that much commonly used for the genome editing. Like in some crops for editing, they have been used in the tobacco, Arabidopsis, and soybean, but they were not as popular as the CRISPR-Cas9, because creating zinc fingers was technically challenging, and the outsourcing was expensive. That's why. Then comes the second generation of the genome editing tools, and they were first used for genome editing in 2011. And you can see here that the talents, they were created by fusing the tail domain. This is the DNA binding domain. These are the tail repeats, which are specific for the DNA. And these tail repeats, they are 34 to 35 amino acid repeats, and the enzyme is the same. So here the specificity for the genomic region is governed by these tail repeats. Then the nucleus is dimerized FOC1, and it creates sticky ends. So talents, they were designed and they can be assembled to target any DNA sequence, but they are sensitive to methylation. So in the DNA, if the sequence is methylated, then talents cannot be used for the cleavage. Okay, so but compared to ZFNs, the assembly of talents was easy, so they were widely adopted for the genome editing methods in plants, in Arabidopsis, in rice, in Brachypodium, in barley, maize, tobacco, soybean, wheat, tomato, potato, and sugarcane. So in many crops, they have been used because their assembly was comparatively easier to the ZFNs. but their limitation was they were sensitive to methylation. So after talents, the CRISPR-Cas9 came, and it is regarded as the third generation genome editing tool. And it was used to edit plant genes in 2013, and it is currently the prevalent gene editing tool. And the CRISPR was discovered as a prokaryotic immune system uh, that protects cells by selectively targeting and destroying foreign DNA. So, in the bacteria, when there is attack of virus or plasmid, so they destroy those foreign DNA. So, CRISPR was discovered as their immune system. So the engineered CRISPR-Cas9, which is used in the laboratories, is based on type 2 CRISPR system, and it has three main components. One is the CRISPR-associated protein 9, that is the Cas9, which has the cleavage capacity. It has two non-coding CRISPR RNAs, that is the trans-activating CRISPR RNA and a precursor CRISPR RNA. So this is the representation of the CRISPR-Cas9. So this is the guide RNA. So this the 20 nucleotides shown in the magenta color is the guide RNA, and this oval structure is the Cas9, and this secondary structure, this you can see in this golden color, you can see. So this is the secondary structure. So this is the CRISPR RNA and trans CRISPR RNA. So the sgRNA combined with the spacer sequence of the 20 nucleotides and 79 nucleotides of this makes it 99 nucleotide long, and this secondary structures binds with the Cas9. So the important point for this Cas9 that it cuts 3 to 4 nucleotide upstream of the PAM sequence. So the PAM for the Cas9 is NGG at the 3' end of the target sequence. So so this PAM is important for the Cas9, that NGG should be there. And this Cas9 has two domains. One is the RUVC domain, and other is the HLH domain. So RUVC domain makes a cut at the strand which is complementary to the sgRNA, and HLH make cut to the this strand. So this is the CRISPR-Cas9 system. The specificity in the CRISPR-Cas9 is determined by the sgRNA. So these 20 nucleotides will determine where it will go and bind. Then the nucleus here is Cas9, the Cas9 nucleus, and the cut made is the double-stranded blunt end cut is made. So this system has been used in large number of species, including rice, wheat, tobacco, Arabidopsis. You can see here many crops, cereal crops, as well as horticultural crops, as well as flower crops. So in large number of species, it was used. And this CRISPR-Cas9 system was much simpler, and it was easy to manipulate than the previously available methods like Jadafen and Talens. And it has high efficiency in producing targeted mutations also. And one other advantage with the CRISPR is that you can target multiple sgRNAs. So multiple sgRNAs can be targeted. So this is known as multiplex editing, that you can do the multiplexing. Okay. So the CRISPR technical simplicity and efficiency made it true breakthrough in genome editing. Okay. So the technique was simple and efficient. So it was used by various laboratories. So this is how this was discovered. So this is the bacterial cell. You can see the Streptococcus bacteria. So whenever a virus, these are the different colors virus is are shown here. So when virus enters the bacteria, this is the viral DNA. It injects its DNA. So if the bacteria survives, it integrates its DNA into these repeat sequences. Suppose when this virus attacked, so this viral sequence, when this green virus attacked this sequence, when this attacked this sequence and this one. So different viruses, different sequences, they are integrated in this in between these repeat sequences. This is known as CRISPR DNA. So, whenever a virus attacks, the bacteria insert a piece of the virus DNA in the CRISPR section of the genome. And between each viral DNA, there is a repeat sequence. So, after that, this CRISPR DNA is copied to make a long RNA molecule. So, this is single stranded. You can see here, this is the CRISPR RNA molecule. This is the CRISPR RNA molecule. So, this is the trans CRISPR RNA which is present in the cell. So when this trans CRISPR RNA is it fits into this CRISPR RNA like a puzzle. When it fits, the Cas9 will binds, and then RNase3 will make a cut. So it will act like a genetic scissor. So when second time when this virus is attacking, so this viral DNA is cleaved by this scissor, because now the bacteria has the memory and it has the scissor. This is the genetic scissor which contain this CRISPR RNA and this the genetic component of this virus. So when second time this virus is coming, so it get cleaved. So this immune system, it works in the bacteria, and using this system, the CRISPR-Cas9 genetic scissors, they were designed. So this is the guide RNA. Guide RNA means that the target chain which we want to edit. So this is the guide RNA, 20 nucleotide long, and this is the trans CRISPR RNA. So trans CRISPR RNA along with the guide RNA, and this is the Cas9. So they will make a cut. Okay. So when the cut is made, then it is repaired. So there are two repair process we discussed earlier. So this is error prone. So so there are chances that nucleotide may get added here, or they may get deleted from here. And this is the template for repair, that if you are giving some template, then there will be no error, and this is the homology direct repair. So so these two women, Emmanuel Charpentier and Jennifer Doudna, so they discovered this mechanism, and for this discovery they got Nobel Prize in 2020 for the development of method for genome editing. So after CRISPR-Cas9, there is another system which is even more efficient than the Cas9. So this is also a third generation genome editing tool, and it was used for plant gene editing in 2016. It has two components. Instead of Cas9, it has CPF1, and it has the CRISPR RNA. So you can so this is the schematic representation of the CRISPR CPF1 system. So there are some differences compared to the Cas9. So you can see here the PAM site is located at the 5' end, and the PAM is different, that is TTN. And the spacer sequence here is 23 nucleotide long compared to 20 in case of Cas9. And the CRISPR RNA sequence is smaller here, only 19 nucleotide long. And this is the CPF1 enzyme. So, we can see here the differences between the Cas9 and CPF1. We can see the difference in the PAM. We can see the difference in the nucleases which are making cut. And here we create blunt end, but here the sticky ends are created. And we can see this RNA difference also, the length in the RNA, CRISPR RNA length difference is also there. So these are the some of the differences between the CRISPR-Cas9 and CRISPR-CPF1. But in nutshell, we can say that CRISPR-CPF1 is even better than the CRISPR-Cas9 system. So because of sticky ends, larger mutations can be created in the CPF1, and the CPF requires a T-rich PAM sequence, which is easy to find, and the CRISPR-CPF1 system is also used for plant genome editing in several species, including rice, Arabidopsis, tobacco, and soybean. So these are the some of the comparisons which we have discussed between the Cas9 and CPF1 system. So if we compare the 4 types of the genome editing tools, then we can say that when we select the target sequence, the options are limited in ZFN, but they are very abundant in case of Cas9 and CPF1 system. The assembly process is laborious in these previous 2 systems, but it is simple in the CRISPR system. And the cost, if we talk about the cost, these systems, they are expensive or very expensive, but they are comparatively cheap. Here, multiplex editing, if you want to target more than one gene, it is difficult in the ZFN and TERENCE, but it's easy and very easy in the CRISPR system. If we talk about the efficiency, it is low and moderate, but efficiency is high in both the systems. If we talk about the off-target effects, they are low. They are low in all the systems. If we evaluate, then the CRISPR-Cas systems or CRISPR-CPF system, they are excellent for genome editing, and they comes in very good and good category. So coming to the application part, so there are many web-based tools which have been developed to facilitate to design the gRNA. So among them, there are CRISPR plant, CRISPR page, chop, where you can give your target gene, and the software will design appropriate gRNA for your target. So these are the softwares which can be used to design the gRNA. Then coming to the applications, what are the applications of the genome editing? So, so the most important application is that we can create the knockouts. Okay, so knockouts can be created, and because NHEJ is the error prone. It can introduce small deletions or insertions, so they can be typically smaller than 100 base pairs, so they can be introduced. And in introduction of indels, it can lead to the frame shift mutations, which can result in the loss of the gene function. And the most important point here in genome editing is that the mutations are stable. Once mutation is created, it is stable and it is heritable in the future generations. So due to its simplicity and high efficiency, CRISPR-Cas is now the dominant tool for knocking out genes. Now, multiplex gene knockout, that it has been used for simultaneously targeting of the multiple genes in the many plant species. So multiple genes can be targeted. That is known as multiplexing is possible with the CRISPR system. So what is the advantage of the multiplexing? That the redundant gene families, which has the related gene functions, can be studied through this multiplexing, which is difficult to study through the normal mutation studies, traditional mutation studies. So they can be studied, and that it helps in the pyramiding also. The pyramiding of the multiple traits can be done. Then we can create a large fragment deletions also. So how it is created? Suppose if we make two double stranded breaks at on the same chromosome at a certain distance, so there are chances that two sites may connect through NHEJ and that intervening sequence will be deleted. There are chances. So here two double-stranded breaks they are created on the same chromosome simultaneously, and which results in the deletion of the intervening sequence. So when you delete the large fragment of the DNA, so it can help in the study of the gene cluster or non-coding RNA. So it has means these applications in this area. So, talents and CRISPR have been used to produce large deletions in rice, Arabidopsis, and tobacco. Then, gene knockout in polyploids. So many crops are polyploids. So, so due to the complex genetic composition of the polyploids, gene knockout are difficult to obtain using traditional genetic approaches. So, gene functions cannot be studied. So the genome editing tools are useful for the polyploid crops also, where the mutant resources are lacking, and using these techniques, mutants can be created. And using the genome editing, the knockouts in triploids like citrus and apple, tetraploids, cotton, pasta wheat and potato, and hexaploids like bread wheat and octaploid sugar canes, they are created. Then gene targeting. So gene targeting is the use of the genetic engineering method to produce one-for-one substitution of a DNA fragment, like gene replacement we have seen, which we are doing in the homology direct repair, or the we are inserting a new sequence at a specific locus, that is gene knocking. So that is possible with genome editing. So gene targeting has many applications in the functional genomics research, like precise gene modifications. Okay, so this gene targeting can be done using genome editing. Then the mutants which are generated through genome editing, they can be directly used for crop improvement as pre-breeding materials. Okay, so they can be used as a pre-breeding materials. Uh, so in case of the tomato, the wild species Solanum pimpernifolium, it was domesticated by editing of the several important genes. So many genes were taken for editing, which were associated with like plant architecture, fruit development. So it resulted in the formation of the new tomato variety or the cultivated form, which has the favorable traits of the wild species. So, nutshell, the undesirable trait of the wild tomato, they were mutated using genome editing, so that the wild has all the desirable traits, and all the undesirable traits they were edited. Then mutations can also be introduced in the coding regions or in the regulatory regions, in the promoter regions, It can be used to generate material to study the quantitative variations. Okay, so it will serve as a mutant source. So in tomato, there are the clevata and bushel genes are there. So these genes, they help in determining the fruit locule number. So the variations, they were created, and the tomato lines were generated with a wide range of the locule number. Wide means these lines, they have mutations in this gene at different positions. So that which is creating the variation in the locule number. So, the quantitative variations have been observed when the gene responsible for inflorescence or plant architecture, they were engineered. Then variations were also observed. Then we can regulate or we can mutate the epigenetic region also. So there are epigenetic modifications in the genome. So to do this, the sgRNA guided fusion protein between we use a different version of the Cas9, that is dead Cas9. So in this, the Cas9, the cleavage function of the Cas9 is switched off, or it is non-functional, and it is fused with the catalytic domain of demethylase. So this enzyme can remove 5-methyl cytosine at specific site. So if in the genome this cytosine is methylated, it will remove that methine. That So it will increase the gene expression. So it will create the epirils. Like we have rils, so it will help in the creation of the mutations in the epigenetic status. So epigenetic mutants are created. So which will help in the development of we have rils. So it can help in the generation of epirils. Okay. So the epigenetic mutant, when we cross with the corresponding wild type, so it will generate the epigenetic recombinant inherit lines. So these individuals from this population, they are genetically identical but epigenetically distant. Epigenetic means modifications which are present on the DNA, like methylation is one of them. So that is different. Otherwise, sequence-wise they are same, but epigenetically they are different. So these type of populations, they have been developed in Arabidopsis, and they exhibit considerable phenotypic variations. So it shows that genome editing is excellent for producing new alleles as well as epialleles. Epialleles, it is talking about the epigenetic modifications, and which are important source of phenotypic variations for crop improvement. Then this is about the basic part. Then suppose if we want to do an experiment which is based on CRISPR-Cas9 genome editing, so how we will proceed? So for that, we have to take a target gene. First requirement, that you should have a target gene with you. So once the target gene is decided, we design sgRNA, single guide RNA. So to design that single guide RNA, we have many softwares which can help in designing the guide RNA. So once the guide RNA is synthesized, then it is checked for specificity, and then it is cloned. So we can do the single and multiplex cloning. Single gene if we have the single gene target, and the multi-gene if we have the multi-genes, we can do. Then after the cloning, we do the transformation to deliver into the host system. And there are methods where we can do the stable genetic transformation using the floral bialistic or agrobacterium mediated. And we can do transient gene expression also in planta. Uh, then after the transformation, we have to screen the transgenics, and for screening also there are various methods like we can do Sanger-based, the NGS-based screening, whether the mutations are introduced or not. We can do the restriction enzyme-based assay, or we can do the real-time PCR. After the confirmation, we do the trait analysis. We phenotypically evaluate our mutants for the desirable characters. So these are some of the vectors which are used for the genome editing. And this is how we select this is an example that we select the sgRNA. Then we clone them into the guide RNA, the cloning. Then after cloning them, we confirm them. And this is the example where the sgRNA for the lycopene cyclase gene in tomato has been shown. So these are the sgRNAs which were designed for the target genes. So these were designed. Then these were cloned into the Cas9 vector. So the Cas9 vector used was the pDirect22C. Then they were transformed, and the edited plants they were generated. This is the experimental approach, that how we proceed when we are conducting the genome editing experiment. Now coming to the regulatory part, that how the genome editing plants they are regulated. So in some countries, the genome editing plants they are not considered as GMOs, like in US and Japan. But in Europe, they are still under regulation. And the difference between the genome editing plants and GMO is that the genome of the edited plant can be free of the exogenous DNA. All the vector sequences which we are introducing, they can be removed in the subsequent generations through segregations. And if you are using like ribonucleoproteins, then the CRISPR reagents, then also the progenies they will be free from the exogenous DNA. So this is the difference between the transgenes and the genome editing, that they can be free from the foreign DNA. In 2013, the first example of the genome editing in horticultural crop was achieved via Italian in Brassica oleracea. But now, in horticulture, several crops they have attained they have been attained using the genome editing, and the most of the studies they have been done in the tomato. Like 42% of the studies are in tomato, and 13% in potato. Okay. And if we compare among the vegetables, floral or medicinal plants, then the 72% of the genome editing work is in the vegetable crops. So till date, around more than 25 horticultural plant species have been successfully edited. And these are the different traits for which they were edited, like the traits which are related to plant growth and development. So, for example, this PDS gene, it gives an albino phenotype. So it was edited in the various species, in like cucumber, in various crops it has been edited. Then the AGL6 gene, which is responsible for parthenocarpy, DELA, and This this gene is responsible for early flowering, then for simplified inflorescence. Then again, the PDS gene in the lilium, and this ACO1 gene for the extended shelf life. So you can see the several genes which were related to the growth and development phenotypes. They have been edited in the various crops. Then these are the genes which are related to the flowering and fruit quality characteristics, like the improved beta carotene, lycopene cyclase epsilon, and the improved hardiness, then phosphorus content in fruits, then CRISPR for the orange fruit, then PSY1 for the yellow fruit, and for the delayed ripening, rin gene, as well as ACS2 gene, and the transcription factor GIP1 for the increased sugar and amino acid content, and this HT16 to modify fruit size and shape. These genes have been edited, which are related to the flowering and fruit quality characteristics. Then there are several genes which are related to the biotic and abiotic stress tolerance. They were also edited, like MR5, then this Quirky genes, then DMR6 gene, MLO genes for powdery mildew resistance, and this NPR1 gene for drought resistance. Many genes for salinity tolerance, like CNH1, then HKT1, then DRAB genes, they were for salinity tolerance. And this eukaryotic elongation factor 4E, it has been edited in many crops for resistance against viruses like cucumber vein yellow mosaic virus, papaya ring spot mosaic virus. So this EIF4E has been edited in several crops for virus resistance. Then the MLO for the powdery mildew resistance. Then this PPO gene for to reduce the browning. And there is huge publications now coming on genome editing. So this is the list of publications which is available in the vegetable crops. So if we see the network of the gene editing methods, so we can say that using these methods, these endonucleases like ZF and CPF1, Cas9, base editor, TELN, we can create a gene knockout, we can create insertion, we can do the replacement, and also we can do the editing, we can create the mutant libraries, we can do the gene regulation. So this we can do. And the process involved the design, then construction, then the transformation, then editing, and then the edited plant. This is the network that how we go through and the processes involved. So if we talk about the regulatory framework for genome editing plants in India, so in India, the plants which were derived from the genome editing techniques, where the site-directed nucleases, they are involved. It can be ZF and TELN, CRISPR, or other nuclease. They are classified in three categories. One is STN1, STN2, and STN3. So STN1 is mutagenesis, where no DNA sequence template is used. In STN2, site-directed mutagenesis using a DNA sequence template, and in STN3, there is site-directed insertion of a gene or large DNA sequence using a DNA sequence template. Okay. So, okay, this is done. So this is the overview that how these three are different. Three techniques are different. So this is the STN1 where cut is made and it is repaired. So this is STN1. So here deletion, insertion, anything can take place. Here a repair is being made through this template. So this template is having the mutant nucleotide. So using this template, this repair is made. So few nucleotides are mutated. This is STN2 category. In the STN3 category, this is this is the repair, but this is the template. Here we are inserting the new element, new genetic element is inserted here. So this is under the regulations like the transgenics. This is STN3, it is under regulations. But in STN1 and STN2, once you prove that your mutant do not have any exogenous DNA, then they are free from any regulations. So so the STN mutants are comparable to those occurring in the nature. Once you remove all the foreign DNA through segregation, then these mutants, they are comparable to those occurring in nature, uh, or the which were obtained through the metagenic treatment or natural variations. So they are similar to those category. STM2 also carries one or several small mutations. So so this also, if you have removed all the sequences, so the edited version could be regarded as allelic form comparable to those available in the primary secondary gene. So this is regarded as allelic form. In STM3 category, because it is template guided, it involve the insertion of a new fragment or new genetic element, which is usually more than 800 base pair at specific locations. So they have foreign gene, so they comes under the regulations like the transgenic plants. Okay, so they comes under the regulations. So now some the crops which were developed, the edited crops which were developed and commercialized. So in 2021, first genome edited soybean was commercialized in the USA, and then it was followed by high GABA tomato in October 22 in Japan. So this high GABA tomato, it contains 5 to 6 times more GABA than the traditional tomato, and it was done by mutating one of the gene that inhibit the synthesis of GABA in tomato, and it helps to lower blood pressure. Then high oleic soybean was developed, where the two genes involved in fatty acid synthesis have been turned off. So the soybean oil from this plant contain 80% higher oleic acid and 20% less saturated fatty acids and 0 grams of the trans fats. Okay. Then non-browning banana was developed. So through genome editing, which reduce food waste and CO2 emissions. Then milder flavor mustard was developed. So through genome editing, the genes which are responsible for bold flavor of the mustard, they have been altered to create milder flavored mustard while retaining the same nutritional profile. So now this leafy green is enjoyed by people as a nutritious leafy green. Then bacterial blight-resistant rice, the genome-edited rice have been developed by disrupting the function of promoters for sugar transporter genes, which are critical for plant susceptibility to infection by Xanthomonas oryzae, resulting in resistance to the bacterial blight. So these are some of the examples of the genome-edited crops. Thank you so much. This is all. Thank you for patient listening. If you have any queries, then you may ask.
Okay, ma'am. Thank you so much. Now I request participants. If anyone has questions, so please turn on your mic or write your question in the chat box. Anyone from the participants, please? Anyone from the participant, please? Okay, ma'am. I think no more questions. Mm, okay. I think no more questions. I think, ma'am, they will understand everything because you are presenting a very wonderful presentation, ma'am. Uh, thank you so much, ma'am. The organizers are thankful to you for taking your time, and also thank you all the participants who joined here. Thank you, ma'am. Thank you so much.
Okay, welcome.